Hello everyone, welcome back to AS and A-Level Biology with Dr. Demi. I am Dr. Demi, and in this video I am going to answer paper 2 questions from May, June 2018. The reason I'm doing this is, besides the fact that I got some requests for it in the comment, it's also, I just think it helps students to get some tips on how they need to answer questions whenever they're faced with different questions. Now, typically students do well in paper 2 from what I have seen, much better than they do in paper 1. Because paper 1, I think, just plays a lot with your mind, especially when you're not sure of what options to choose. Two things look correct, so it might be a bit daunting. But paper 2 is more straightforward because it's structured questions, so it's easy for you to be able to just answer those. But still, there's some tips that would help you as we go along. If you have just found this channel, I am posting the AS and now A-level content in chronological order. So you can search out the chapters that you're looking for, chapter 12, 13, 1, 2, whichever one you're looking for, you'd find all the videos under that chapter. And so you'd be able to then follow chronologically if you're trying to revise for the exams, or if you're just trying to get an understanding of the content before you go to class. This is also a good resource to use for that. Okay, so again, for this video, I just randomly picked questions. I did not necessarily just go through the paper, skimming through, trying to answer every single question. So these are just a few questions, and we'll see how these videos do with you guys. If you find them helpful, then perhaps I'll be able to post other videos so that you'll be able to see how I would want students to answer questions like this. All right, so let's have a look here. This says sucrose is a disaccharide, and this figure over here, so let me just use my annotation tool. This figure here shows how sucrose is broken down in a reaction with hydrochloric acid. So if we add hydrochloric acid to sucrose, we would then get these two components over here. All right, so the question then says, name the products A and B shown in figure 2.1. So product A is glucose. This is what glucose looks like. And I'll tell you just something just in a bit. This is glucose, and this here is fructose. Now, if you're not very observant as a student, you might make the error of seeing this as glucose. I know some students have seen it and they thought, oh, maybe this is some other special arrangement of glucose. No, it's not. This is fructose, and fructose is the one that looks like it has like a roof on top of it, sort of, if we want to look at it that way. And glucose, on the other hand, has a flat top with an oxygen, and everything goes on. If you watched the chapter 2 videos that I did, if you haven't, please do so, because they'll be very helpful in answering a question like this. You would know that this is alpha glucose. So even if you say alpha glucose, that would also be marked correctly. But B is certainly just fructose. We don't know if it's alpha fructose or just fructose is fine. All right, then it says to name the type of bond that is broken in this reaction. So this is the bond that you're breaking over here. And again, if you watched the chapter 2 videos where I spoke about polysaccharides, disaccharides, and so on, you would know that this is a glycosidic bond. Now, for some reason, students tend to find chapter 2 a little bit confusing in terms of knowing which bonds belong where. And I often tell them that when you think of glucose, when you think of carbohydrates, generally, just think of glucose. Glucose is sometimes written as glyco, or just think of glycogen. It has something to do with glucose. And so you would then be able to remember that the bond between carbohydrate molecules is called the glycosidic bond. Between protein molecules, it's called the peptide bond, which makes sense because the primary structure of a protein is a polypeptide chain. So that would be the peptide bond. Then it says here, state the type of reaction that's shown in figure 2.1. Wherever you have the addition of water to break a bond, because you can see here, or rather wherever you're breaking a bond, that would be called a hydrolysis. Okay, so this is a hydrolytic reaction. So there you would just write hydrolysis. Then question B here says that when Benedict solution is added to a sucrose solution and put into boiling water, no color change is observed. And I just need to point out here that for many students, the food tests, like the way you test for glucose, fructose, and not fructose, glucose, lipids, proteins, and so on, Many students tend to think that is limited to paper 3, but it's not. You usually get questions about food tests even in paper 1, or you can get them in paper 2, as you can see here. So over here it says when you add Benedict's to a sucrose solution and you put it in boiling water, in a boiling water bath, you don't see a color change. And the reason for that is that if you remember the test for sucrose, which is a non-reducing sugar, I believe, yeah, sucrose is the non-reducing sugar. Sometimes I confuse which one's non-reducing and which one's reducing. But anyway, if you have sucrose, whenever you want to test for sucrose in the solution, you would usually add some kind of acid to it to break the bonds between the sucrose molecule. And then you add the Benedict solution and then put it into boiling water to check for a color change. If you don't add the acid to break the bonds, you will not see a color change because you haven't released the glucose molecule to be tested. So you need to break the bonds in order to test for the glucose molecule, to test that this is sucrose indeed. And because this is 2 points, I would say that you have to give 2 things. So state why no color change is observed. First thing you have to say is that the bond between the sucrose molecule has not been broken by the addition of hydrochloric acid. That would definitely give you 1 mark. And then your second mark would then be the fact that what's it called now, that you don't have any reducing sugars present. So glucose is the reducing sugar, sucrose is the non-reducing sugar. I think I'm sure of it now. So for you to test a non-reducing sugar, you have to first of all hydrolyze the bond. If you do the Benedict's test directly, you're not going to find any reducing sugar because there is no reducing sugar that has been released. You have to break the bond in order to release glucose, which is a reducing sugar, and then get your test. Fructose is also a reducing sugar. All right, let's look at an enzyme question. So over here, this question says that researchers isolated a sucrase enzyme from the bacterium Bacillus subtilis. They immobilized the enzyme in alginate beads. I hope you've learned how to do that at school. If you haven't, please ask your teacher to tell you how to do it. The researcher investigated the effects of temperature on the activity of the immobilized sucrase compared with the activity of the same enzyme free in solution. So let's just take note here. This is the one that's free in solution, and the X's are the immobilized ones. And the question here says, compare the effects of temperature on the activity of the immobilized sucrase with the activity of sucrase that's free in solution. So here, I think what I would say to you here is that try to, first of all, look at what the overall trend is for both reactions, or for both types of enzymes rather. In the immobilized one, here it goes high up from 40 degrees as temperature increases, and then it continues on a downward spiral, but it's still active if you want to think of its activity until it gets to around 80 degrees. Whereas with the one that's free in solution, it sort of has a similar trend, although you can see that it's not necessarily as high as the immobilized enzyme, and it tends to lose its activity quicker. So what you would then say here for 4 marks, you would have to say about 4 different things. So first of all, you have to say the activity increases up until a certain point and then decreases. The certain point here is around 60 degrees Celsius. You then also have to say that both of them have an optimum temperature of 60 because it seems that that's where they both peak before they start to go down. And you would then also have to mention that the enzyme that's free in solution denatures quicker, loses its activity faster because it's free in solution and that's after you've reached the optimum. Whereas with the immobilized enzyme, the activity seems to be higher. Even at a very high temperature of 80 degrees, there's still some level of activity for that enzyme. So if you're able to state all of these things, you will get maximum marks for this question. All right, let's look at number 4. Like I said, I just picked these questions randomly, just by the way. So the fact that they just seem to like be chronological, that's just a coincidence, because I was just taking screenshots randomly and just saying, okay, I think this might be a bit difficult for students, so let's have a look at it. Okay, let's look at this one. I like questions like this because they're just really easy ways for you to get full marks. So here it says, state the term that matches each of the descriptions given in the boxes E, F, G, H, and J in the figure 4.1. So here it says E, so let's start with E because it's right at the top. It says and first of all, the question says this is a ribbon model of hemoglobin. So it means that when you're looking at this and you're trying to answer all of these terms, you have to have hemoglobin in your mind. Otherwise, you'd answer things that would technically be correct for any enzyme, but might not apply to hemoglobin. So always make sure you read all the instructions carefully so that you know the context in which you're answering the question. So here E says a chemical group that is not made from amino acids. So if you remember when I discussed chapter 2, and I was talking about proteins, we discussed the structure of hemoglobin, and that is the fact that it has 4 globin chains. Those are 2 alpha globins and 2 beta globins. And it also has what we call a prosthetic group or a heme group. And the heme group is not made from any amino acids. It's just a prosthetic group that allows hemoglobin to carry out its function. So E here would be that prosthetic group, or you can say it's the heme group, and I'm pretty sure that would be correct. Not pretty sure, I'm very sure that would be right. And then F here says a form of secondary structure found in the polypeptides in hemoglobin. So, I mean, just by looking at it, this is not something you really have to stress about and say, oh my goodness, what is in hemoglobin? You just need to look at the structure itself, and you can see that this is an alpha helix. All right, so this then would be an alpha helix. Please, when you're writing an alpha helix, please make sure you write your alpha this way. All right, don't write A. Okay, if you don't want to write it this way because you're not sure of how to write it correctly, then write it out as alpha helix. Okay, don't helix is two alpha helix is 2 words, it's not 1 word. So don't write a helix because that would be marked wrong. So you either write alpha this way. Okay, so you either write alpha helix or you write it in 4 words, and that would be correct. Let's look at G. Each polypeptide is folded to form a complex 3-dimensional shape. So if you think back to the structures of proteins, remember that we have 4 levels of structure. We have the primary structure, which is the sequence of amino acids. We have the secondary structure, of which there are 2 types. That's the alpha helix and the beta pleated sheet. And then we have the tertiary structure, which is the folding to form a 3-dimensional shape. That means that G would be referred to as the tertiary structure over here. Something else I also feel the need to tell students, and I see this with my students a lot, please don't write the short hand form of things. So for example, students will write things like TERT structure. This means nothing. Please write out tertiary. The time that you save from writing TERT, you're not going to use it for anything spectacular, except you're planning to save the earth from an invasion. So please make sure that you write things out correctly. As you should. So H here, each polypeptide is made of a sequence of amino acids. I've just said it. That is the primary structure of the protein. And J says each hemoglobin molecule is composed of 4 polypeptides, shown here by different shading, and that tells you that it's a quaternary structure. So J would be quaternary structure because it's just telling you that this has 2 or more polypeptide chains, and that is the definition of a quaternary structure. Okay, let's look at this question. So this question is also from the Major paper 2. This is the 2018 paper. And this question is asking us, I think, just looking at the graph, I can tell it's something that has to do with transport in mammals. So let's read the question. The effect of carbon dioxide on the percentage saturation of hemoglobin with oxygen was investigated. A sample of mammalian blood was exposed to a gas mixture that contained increasing partial pressures of oxygen. Throughout the investigation, the partial pressure of carbon dioxide was maintained at 5.3 kilopascals. The percentage saturation of hemoglobin in the sample was determined as the partial pressure of oxygen increased. The investigation was repeated with a second sample of blood in which the partial pressure of CO2 was maintained at 10.7 kilopascal, and the results are shown in the figure. And the question then says, use the information in figure 4.2 to state and explain the effect of increasing partial pressure of carbon dioxide in muscle tissues during strenuous exercise. So here, This is an interesting question, first of all, because they're asking you for quite a variety of things here, and there's so many ways to answer this. But it takes me back to chapter 8. So if you haven't watched that video where I explain what is called the Bohr shift, I'm just going to write it here. Please go have a look at that. The Bohr shift simply says that whenever you have carbon dioxide present in the blood, and you also have oxygen, hemoglobin for some reason has an affinity for carbon dioxide as well. So it doesn't only have the function. So hemoglobin's only function is not just to carry oxygen. It also tries to remove carbon dioxide from the blood. So whenever carbon dioxide is present, hemoglobin will have a tendency to want to bind to that carbon dioxide so that it can process it, remove it from the blood, and resume its activity of binding to oxygen. However, what that means is that as you increase carbon dioxide concentration, you would find that even though there is an increase in partial pressure of oxygen, the percentage saturation of hemoglobin with oxygen would not be as high. So in this case here, when the partial pressure of CO2 is at 5.3 kilopascals, you can see that let's try to do some reading here. So the partial pressure of oxygen here is 2, and if we just read directly from 2, I made a little blunder there. If we just read directly from 2, we will see that the percentage saturation of oxygen, I'm trying to read on the very first graph, so the one that says 5.3 kilopascal. So the percentage saturation of hemoglobin with oxygen is about 22, all right? But now if you look, when you increase the concentration to 10.7 kilopascals, let's look at what happens here. I'm still reading at a partial pressure of 2. Then the percentage saturation of hemoglobin with oxygen is about 14%, simply by increasing CO2 concentration. So what that's telling you is that hemoglobin is dissociating from oxygen and binding to CO2, and that is what we call the Bohr shift. So the Bohr shift would definitely be an effect of CO2 increasing in muscle tissues during exercise. You would also find again that the percentage saturation of hemoglobin would decrease. Something else that you might want to mention here is that when you have the Bohr shift, you would also have an increase in the production of carbonic acid in the body. And when you have an increase in the production of carbonic acid, it means that you have more hydrogen ions in the blood, and you can also say that the affinity for hemoglobin between hemoglobin and oxygen would decrease. Now, because this is just 4 marks, you don't have to say more than 4 things. So I often tell students, because this is something else I notice with students, so often because they are unsure of themselves when they answer certain questions, they tend to go above and beyond and explain things that don't necessarily have to do with the question. I've even had students who draw extra lines under the question just so they can keep writing. So I have to say this, that biology is not an essay, all right? You need to be very confident in your answer. So simply state things as you are asked to, and don't exceed the space that has been provided, because it's often not necessary to do so. They've already decided that they've figured out that that would be enough room. And so to save room on questions like this, something I always say is don't start with an introductory statement, because that's something else I see students doing in the bid to sound very well put together with their English and their sentence construction. They would start by, first of all, using the first 2 lines here to say something like the effect of increasing CO2 in muscle tissues would be as follows. There's no need for you to do that. They already know what they asked you, so just answer the question. There would be the Bohr shift. The Bohr shift has been observed, especially with an increase in concentration or increase in partial pressure of CO2. The affinity between hemoglobin and oxygen has reduced, or would reduce rather, because you're stating it in terms of the muscle tissue during exercise. There'll be an increase in carbonic acid production in the blood because of this as well. And then you would also say partial pressure or percentage saturation, of hemoglobin would decrease with increase in concentrations of CO2. Okay, let's look at this question. So I picked this question specifically because students tend to not like transporting plants, and I don't know why. I feel like it's one of the really easy chapters. Yes, indeed, you can't relate because you're not a plant, you don't have xylem vessels or phloem cells or whatever it is, but it's really easy to study the diagrams and know what's what and where. So I would encourage you, please don't be discouraged by it. I did a video on this chapter already. It's chapter 7. So you can just go have a look to see how the chapter looks. I can promise you it's not bad, and it's also not long. So it summarizes everything for you really nicely. All right, let's look at this question. It says water absorbed by plant roots travel by different pathways from root hairs to the xylem. And figure 6.1 shows these pathways in the root of that plant. I'm not going to attempt to say that. So let's just look at the different pathways. We have pathway B and pathway C. So pathway B there, the ones with the straight arrows, you can see them over there. And pathway C has a lot of dotted arrows. So actually pathway C is quite common over here. And then it says name cell A and pathway B. So cell A in this case would be the endodermis. And I know that because I know how the structures are arranged, and I think this is also highlighted in your textbook where it discusses the movement of water from root hair to xylem, and you would find that the water keeps moving until it reaches the Casparian strip and then follows a different pathway until it is able to enter the xylem. So that's just something you need to sort of learn. Please try to, I don't like telling students to memorize, but in this case, I feel like it's helpful because, like I said, many students can't relate to this content because it has to do with plants. So cell A is the endodermis. Pathway B is the apoplastic pathway. The reason I know this is that when I look at pathway C, I see that the water is moving through the plasmodesmata. So the water is moving through the different linkages between all the cells, between the cells that make up this plant. Whereas with pathway B, which is this one over here, the water seems to be heading straight for the cell wall, and that is what the apoplastic pathway is, where water moves through the cell wall into the plant, and it goes from cell wall to cell wall. So that would be the apoplastic pathway. Then this question, the B part says, transpiration occurring at the leaves is mainly responsible for movement of water across the root of the plant. Explain how transpiration is responsible for the movement of water across the root as shown in this figure. So I'll try to explain this to you using a drawing of a root or a plant cell, which would not necessarily look like a plant cell, but drawing is not something that I was blessed with, but as long as you get the picture, I hope it's fine. So here I'm just going to write H2O abundant. All right. And then over here, I'm just going to draw things that, well, you can imagine are leaves on the plant. So a good way to think of this question would be, first of all, to ask yourself, what is transpiration? Transpiration is the loss of water from the leaves, right, or from the aerial parts of a plant. So in this case, water is being lost to the atmosphere. And what that means is that there would be a concentration gradient because water is lost from the leaves to the atmosphere, and you then have over here water in the roots that are abundant. And what that would cause is that it would result in a concentration gradient that then causes water to want to move into the roots. So water would diffuse into the roots, okay, and start to move up into the plant. And what this simply means is that transpiration, first of all, creates a concentration gradient between the roots and the leaves. Transpiration also causes what we call a transpiration pool, which means that as water is lost into the atmosphere, there is more uptake of water because it's more like osmosis, trying to keep things in a state of balance. And so water would keep moving up through the roots through the roots into the plant. We also have what we call the tension in the xylem vessels. So the xylem vessels are arranged in such a way that they're able to allow that tension of water to cause a column of water to flow. You can also refer to the forces of cohesion and adhesion, and how the transpiration, so the loss of water from the root, would cause the water molecules to move in a single column because they're trying to balance things out. So the adhesion as well as the cohesion between water molecules and the hydrogen bonding between the water molecules would help. Again, for this question, because there are 5 points, please just mention 5 things and try to explain them just like I have just now. Like, loss of water from the leaves creates a water concentration gradient. Transpiration will result in the transpiration pool. As a result of a transpiration pool, there will be movement of water in a single column due to the hydrogen bonding between water molecules. Forces of adhesion will allow water molecules to move into the xylem successfully, and forces of cohesion allow the water molecules to also stick together and move well. The xylem vessels also allow for some form of hydrostatic pressure that allows the water to continue to move as transpiration occurs. So I hope that explains it to you. Again, don't exit the space that has been provided. Don't write 10,000 things. Be very confident in your answer so that you can get maximum marks. And don't use an introductory statement. So don't say transpiration is responsible for the movement of water by doing the following. Don't start with that. Just go straight into your answer, and you are sure to maximize your space and your time effectively. This is the last question that I chose from this paper, and I hope that the explanations have been helpful in, just getting you ready for the exams or how you approach questions when you're answering them. I'll try to do this more intermittently, but I want to also record chapter 14 of the A-level biology syllabus. So please check out all the AS videos if you've started preparing for the major exams. Check them out, study them, just give them a good try, and make sure that you're practicing past questions, not just by yourself, but also with friends, with colleagues, so that you can improve your understanding. And don't forget to check out these videos so that you can revise or even check them out before you go to class to learn something new. Thank you all so much for watching, and until next time, have a good time. Goodbye.